Okay, so welcome to our 27th lecture. So today I'm going to introduce you to another omics, and this time is phenomics, and how we how this omics kind of focus on phenotype, like the outward appearance or disease status, and how this link to all the other omics that we know, and what are other kind of ways that we can collect and analyze data in biology. So today's agenda, we're going to talk about what phenomics is, and you might be surprised that in many review papers, people are still not sure what constitutes phenomics. So it's not that clear, but basically it's a focus on phenotype, but it's similar to other omics, but we can see some uniqueness in there. So we're going to talk about some ways that we can profile the phenotype in a high throughput fashion, so that we can apply computational analysis. And we look at some application in biology, in medicine, in agriculture as well. Okay, so let's start with what is kind of phenomics. When we think about the pathway from the cells, like inside our body, inside the living organisms, from the genome sequence to gene expression, to protein expression, to the metabolite level, these are what we covered in this class before. But everything here kind of together create the phenotype, or the appearance, or the function that we see in the organisms. And one thing that people who study phenomics also include is the impact from environmental factors. In this class, up until now, we focus on everything inside the cell. We focus on everything molecular. This class is computational molecular biology. But we know that when we look at the disease, look at the phenotype, look at the maybe the morphology of the human, animals, and plants, it also depends on the environment factors. So if I were to define phenomics, I try to combine definitions from multiple opinions. So it's an omics, so it's a high throughput. It's a high throughput. It's a high throughput characterization of phenotype, and the dynamics of the phenotype, and the combination of phenotype. So this is different from the usual biological research where you might do multi-omics, you might get the mutation data, gene expression data, protein data, but you're usually focusing on just one disease or one function of the organisms. So you are only looking at one phenotype at a time, mostly. But for phenomics, they want to recognize that there are multiple phenotypes that you can see from an individual, from a cell, all at the same time. We want to analyze every phenotype together as a combination, and also to include the impact of environment on the phenotype as well. So let's look at some more examples to give you a clearer picture of what phenomics might be. So in the context of medicine, for example, we may think of the phenotype of a patient might be something that we can get from clinical measurement. We can get the blood pressure, we can do a CT scan, X-ray scan, we can do molecular tests as well for mutation, for biomarkers. So when we think of a person, usually in a biology project, we pick one phenotype, maybe the biomarkers or maybe one disease, and we study all the molecular aspects of that disease. But on the other hand, on the phenotype side, we can have multiple measurements all at the same time, about the health status of the patient. So phenomics, they want to understand every phenotype together as a single description for a human. And the idea is once we combine the idea of multi-omics, molecular data that we have, against all the phenotypes that we can collect, we have multiple variables on both sides. Hopefully, this will allow us to identify the cause of the phenotype, the cause of the disease, and which treatment could be used on the molecular level to treat the phenotype that we see on the outward of the patient. So we want to study both the inside, the molecular level, and the outside, the phenotype. If we think about phenomics in, let's say, in animal, in livestock, the phenotype of animal in the farm might be their health status, and also production yield, how much meat they produce, how large or how many children they can produce, how many eggs they can produce. So it's slightly different from human. For human, it might be mostly about medicine. But in animal, we also focusing on their morphology, like their size, their production. And the difference that we're going to see is that in livestock and in agriculture, we have more control over the genetics. Like we can do breeding, and we have more control over the environment. Like we can control their food, we can control their habitat. So we can control more about the environment. But compared to human, like in medicines, we usually cannot control how that person was raised, like or the food or the culture. So usually those are confounding factors. But in animal and in plant, like we have more control. So we can do phenomics in a more precise, like more controlled manner. And we have more ability to monitor as well. Like for human, you might violate the person's right if you monitor the activities like all the time. But we can do video real-time monitoring for animals and plants. So this allows us to use the idea of phenomics to eventually optimize breeding, optimize food for the animals, optimize nutrients for the plant, and optimize the whole production system. So in animal, we might rely on videos so that we can see the movement, maybe the activities, and we can monitor how much they eat. We can track individual animals in the farm. For plants, because plants are stationary, so people can do different type of monitoring. We can grow plants in the field. Each field might have different nutrients, different level of fertilizer, and we can take aerial image using drones, and we can analyze the maybe the different breeds, different cultivar of the plant combined with different nutrients, and look at the phenotype, maybe the yield of the rice, or maybe the density of the plant. And for plants, because they are stationary, and we can grow them in larger numbers, we can rely on visual-based phenotype as well, like the color, the shape, or the weight of the plant, and different part of the plant as well. The leaves display different phenotype. The root might respond to water and nutrients more strongly, so we can see more visual phenotype as well. And we have more control over the experimental options. In animal, you might not want to introduce disease intentionally, but in plants, we have some model system that we can introduce disease, introduce other factors. Okay. So, up to now, we see that phenomics is the focus of phenotype. But when we want to explain the phenotype, we also understand the molecular mechanisms. So some people kind of view phenomics as related to system biology. Actually, they are complementary. So when we learn about system biology, we learn about system biology as the pursuit of knowledge. If we know all the factors that at the molecular level that explain certain function or certain phenotype of the cells. So we are looking at the bottom-up knowledge. We want to understand what causes such and such phenotype or function. The ability of the bacteria to digest certain compound, maybe we want to understand which gene or which enzyme can take part in that. But for phenomics, it's in the opposite direction. We want to understand the phenotypes of the organisms of the bacteria, and we want to understand, not only to understand, but we want to manipulate them as well. We want to change the phenotype. We want to make ourselves healthier. We want to make the plants yield more. How can we do that? We still need system biology to understand which gene, which metabolite contribute to the yield of the animals. But we are looking at the manipulation aspect as well, not just the understanding, but we want to change the phenotype. How can we change it? What are the tools that we can use? So looking at another way, so system biology, we want to understand from genotype down to phenotype. We want to understand how genotype and environmental factors together work to produce the phenotype. We want to know the mechanism. But for phenomics, we want to understand the change, how to affect which nutrient should we increase or decrease in order to affect the growth or the health of the plant. We want to know we have to have the ability to manipulate. But the ability to manipulate kind of comes from the understanding that we get from system biology. So just to summarize, the reason why we want to look at phenomics, at least a little, is we want to, for application, we want to change the phenotype. So we want to understand how molecular change propagate to create different phenotypes. But when we study system biology, we want to understand the molecular mechanisms. Usually we kind of either control or ignore the confounding factors, environmental effect outside of the cell. But for phenomics, this is an important factor because you can change the fertilizer, you can change the growth condition, and you change the phenotype. So this becomes more important compared to in system biology. And it's a focus of application. In personalized medicine, we want to understand how we can improve maybe the blood pressure or maybe reduce the weight of a person, or in breeding, we want to create better, more resistant livestock and plants. So I would say that phenomics and system biology kind of complement each other. If you know both, if you study from both aspects, you get a complete picture of an organism. Okay, so let's look at what the components are in phenomics. So the first part is the phenome, the omics of the phenotype. So if we want to kind of classify kind of roughly what are the phenotypes that we can measure, we might think about the kind of the appearance based or physical based phenotype, like morphology. For human and animal, like we have the weight, height, maybe the shape of the body. For cells, like we saw in the microscopy lecture, that we can measure the size of the shape of the cell, like the ratio of the nucleus to cytoplasm. So these are phenotypes that we can see by eye, by microscope, or by imaging. The other class of phenotypes are behavior, these things that change over time, or for example, such as the feeding behavior, or the active and inactive time that a person might spend every day, or the animal might spend kind of every day. These are behavior that we can monitor, usually through maybe wearable device, or some sensors, or some videos, or using video camera. The other one would be the one that we are more familiar with, the metabolic and molecular phenotypes. So this one we collect using omics technology or functional assay. So basically today we are going to kind of expand our thinking of data collection and analysis from just molecular level to now something that are outside kind of the cell as well, at the organism level. So to make sure that like different labs can measure different things about the animals, about the person, about the plant. So to make sure that data from different studies that are compatible with each other. When someone says that, okay, I'm going to collect the eye colors. If you use different words like for the colors, that can be some discrepancy. When you collect the shape of the plant, there have to be some standards about which part of the plant, at which age, at which time of the day, that affect the body of the plant. So people, similar to when we learn about functional annotation, like gene ontology, we have to have standardized terms that characterize these different phenotypes. So for human, there is a human phenotype ontology. So similar to gene ontology, this will consist of different terminology that are related to human disease or human morphology. And there are some relation, like if you have abnormality of the organ, like it might be in the muscle system or might be in a different system. So these will be kind of related to each other as a network, similar to gene ontology. So human phenotype ontology they start off as only looking at hereditary disease, like genetics disease, but now they also include generic clinical description as well. So similar to when you go to hospital and you get diagnosed, that there are standardized terms for the disease, standardized terms for your symptoms. So if everyone use the same standard when we can combine the data together. So this study, usually they will go and analyze all the medical texts. They will look to publication database and take extract the description of disease, maybe the association between disease and gene from literature, and then they analyze the association between if this disorder is mentioned in the paper, what are the words, what are the keywords that people use to describe them. So these are the only way that we can kind of standardize how different people around the world describe the same disease. For other systems like animals and plants, there is this open biological and biomedical ontology group that try to kind of collect opinions and the convention that laboratory around the world use to describe animals and plants. So these, if you go to this database, there will be different ontology in there. Like for example, this Drosophila phenotype ontology. So every phenotype that we induce by mutating Drosophila, or you might see the plant anatomical ontology. So people who study plants have standardized terms to describe different parts of the plants and what are the phenotypes that they measure. So these are community-based. So this is based on people around the world who study the same animal, the same plant, come together to propose how they standardize the way that they describe the data. So it's kind of facilitate data integration across labs. Okay, so let's look at some phenotypes that we can measure. So this will be in contrast to molecular level phenotype that we learn throughout this course. So this lecture might be probably out of the topic of molecular biology a little bit, but it might be useful for you in real world. So we have seen using microscopy, we can extract image-based characteristics of the cell. What are the protein expression? If protein expressed in spot, how many spots there are, how far are they from the nucleus? We can describe the expression level of gene and protein inside the cell. We can apply the same techniques to animal and plant. Using the technology that we have nowadays, we can detect individual animals in the image. We can detect different parts of the plants, different leaves, automatically using computer vision, using artificial intelligence technique. So this allows us to look at multiple plants at the same time, track multiple animals at the same time. So this allows us to get morphological measurement. We can measure the surface of the plant, of the leaves. We can measure the length of the stalk. We can measure the color of the leaves. So we understand multiple phenotypes with just one image. With videos, we can also track how much the animal moves around. We can even calculate the distance that they move in a day and how much time they spend lying down or walking on. So these are behavior phenotypes that can tell us about the health and status of the animal. So again, just to give you more details, using modern day AI, we can detect not only that this is a cow, but we can also detect that this is a sitting cow, so a walking cow. And for walking animal, we can also measure the pose of the animal, how much, what's the angle when they move their leg. So similar to in medicine, you might have heard that several hospitals now have camera that if you walk toward the camera, it can detect how you walk and determine if your balance is good or if you might have a certain risk of stroke or not. So with modern day technology, we can identify the movement of body parts, and then we can use that to develop detailed phenotypes to characterize if the animal is healthy or if it's limping on its leg, for example. And in medicine, if you ever study radiology, so when we take imaging of human body, we also want to understand if there is any patterns inside the scan that can tell us the severity of the disease. Maybe it can tell us if the patient has certain mutation to cause certain cancer. that appear as certain patterns inside the image. So this is similar to when we talk about an image of the cell, and we calculate the shape, and we calculate the density of the cells, we calculate the texture of the cell. So people in radiology also apply the same similar techniques. From the scan, they extract the position of the tumor, and within this position of the tumor, they want to understand the physical characteristics. They want to understand if the tumor is rough or if it's smooth, how large is it, like how loud is it. So they calculate the morphological parameters, the intensity of the color that they get on the tumor, and the texture that they get on the tumor. And then we can use these kind of thousands of phenotypes, basically phenotypes of the human, of this patient, to identify if any of those signatures can correlate with the disease or maybe correlate with the severity. So nowadays, you might have seen more and more of a database that covers multiple phenotypes. This phenomic database, like the UK Biobank, it not only includes the medical clinical measurement like the MRI, blood pressure, bone scan. Now they also try to include environmental information. They have the demographic of the patient. They have the ethnicity of the patient. They have the lifestyle information of the patient. And they also try to integrate hospital record. If the patient has certain disease before, that can explain the current phenotype, that can explain the future phenotype. So these databases try to connect what we usually have from the medical side, like the clinical measurement, and what we usually have from the molecular biology side, the omics data, also together with with environmental information. So they collect these maybe through questionnaire, like they ask the patient how much you eat, like how much you exercise. So these external factors can contribute to the explanation of the phenotype. They might ask the patient to wear some Apple Watch to monitor the activity or monitor the heart rate to get more information about the behavior and the external factors about the patient. On the US side, there is the PheWAS project that tries to combine medical records with DNA sequencing, so the molecular and the clinical side, but also together with the behavior information and to fit it, like wearable device or to survey response. So nowadays people start to realize that knowing the omics alone is not enough to explain the phenotype because there is also impact from external environmental factors. Clinical data alone is not enough because it only measures the phenotype. Clinical data doesn't include the molecular aspect that explain phenotype. So this database tries to combine all three, the phenotype from clinical measurement, the omics data to gain molecular knowledge, and the environmental information through wearable device and lifestyle questionnaire to together understand how these three factors affect the phenotype. But one thing that's very important that limit our understanding of phenotype in the past is that people might not pay too much attention about the metadata. Like for example, like when you analyze an omics data, sometimes we talk about what is the sample size and how many patients do we have. But we have to be careful that if the patients come from different countries, if the patient have different age, or the patients might have a different states of the disease. If you combine those data together, it's going to confound your analysis because the association that you found might be because patients from developed country might have certain lifestyle that can affect their molecular or their behavior or their phenotypes. So metadata is the details about how the data is collected. And that is important to control a lot of confounding factors. Like for example, if you want to study plants and insects, seasonal and climate information is very important because plant growth and insect growth depends a lot on the seasons. But if you are studying human and animal, seasons might not matter much because we are the same, almost the same all year round. When we collect data in the lab, if you use different protocol in the metagenomics lecture, we learned that if we use different DNA extraction protocol, or if we use different 16S hypervariable site, we can get different bias to a certain bacteria. So the details about how the data is collected, how the sample is prepared, and what platform is used, also is a confounding factor to the data analysis. So in the past, these are not carefully collected because in for example, in clinical trial, they try to control for all these factors by using inclusion and exclusion criteria. So there is no strong confounding effect inside the study. But nowadays, that we have so many data sets from different populations, from different maybe genome sequencing platforms. If we are not careful about the details, about the metadata, we can risk getting wrong knowledge, incorrect knowledge, because we merge data that are not compatible with each other. So one of the goals of these modern phenomics database is they want to be very complete, very thorough about the metadata. What are the information about the patients, information about the data that they collect. Okay. So this is the first part. We now know what the phenome is, the collection of phenotypes. So now let's look at the high-throughput technology that we can use to obtain phenotype data. So for the molecular information, we have omics technology. So we already know this very well. I'm going to skip it. But what we are going to talk more about in this lecture is how you use imaging, how you use robotics or drone or other technology or wearable device to get more detailed phenotypes about human, animals, and plants. So let's look through some examples. So let's say you want to study, if you will go the forest, you want to keep track of how the trees grow. Does it meet the original density or the general height that you want to achieve? So in this case, let's say you use a drone to fly over the forest area. You take the image of the trees and you try to extract information. So in this case, might be the height of the tree. We want to understand how fast the trees are growing. So we have the height information over time. The reason why I picked this example, because it reminds us of the idea of spatial transcriptomics. In spatial transcriptomics, we have the tissue, and we know that on the tissue there are many cell types that together form the tissue structure. In this case, you can think of the forest as the tissue. And inside the forest, there will be local densities of certain type of trees that grow well together. So we have different biomes on the map. So in this kind of study, when they get the phenotype of the tree, the height, maybe the colors, they also want to make sure that their information can classify the type of the trees or the type of the biome inside the forest. So they use the same idea as transcriptomics, spatial transcriptomics. So basically, they look at they separate the map into grids, and in each grid, you have different phenotype of the tree. One thing that you can do, similar to in single cell data, you can cluster these grids together based on the similarity in the plant phenotype. So some grids that have the same tree, same height, will be more similar than different grids that have different type of trees, taller or shorter tree. But you also want to constrain it so that grids that are next to each other, like biomes that are next to each other, should also be similar. So similar in the tissue structure. when you infer the tissue structure, if you say that this is a infiltrating immune cells, you expect the immune cells to be occupying certain region, not in tiny spots throughout the tissue. So they use the same technique. They combine similarity in the phenotype to the similarity to the kind of adjacency on the map to classify forests into different biomes. So we use the same techniques, but instead of on spatial transcriptomics, we use it on the phenotype that we get about the forest. So this is based on just regular imaging, just photos. From these photos, you can reconstruct the height of the tree and the colors of the vegetation. But imaging is not just about taking photos. You might have heard about spectral imaging before. So using different wavelengths, you can image different aspects of the organisms, usually plants. If you pick the right wavelength, at certain wavelength, you can see the response of the plant to loss of certain nutrients, for example. So there are many technologies that we can use to study different phenotypes. With videos, we can do motion tracking. Let's say you have a farm and you establish your camera several positions inside the farm. So what we can do with kind of modern technique is we can recognize individual animals. We can track how the animals goes from one corner of the farm to the other corner when that animal change from one camera to the next. So we can measure how much the animal moves around by mapping the position that we see in the camera back on top of the model of the farm coordinates. So we can calculate in details the activity of the animal. For plants, because it's stationary, so people usually use a time-lapse image. You don't need video because plant doesn't move around for you to track. So you can keep your camera stationary, the plant stationary, and takes an image maybe every 1 hour, every 3 hours, for example. So this allows you to track the growth of the plant from one time point to the next. Maybe it grows more leaves, maybe the leaves get larger, and once you maybe you give it more fertilizer, or maybe you remove the fertilizer, you can see how the plants change. The color may change, or the size may change. So nowadays we have kind of software library that allow you to put in this time-lapse image, and it will extract the shape, the size of the plant, the color, and give you a visualization of the change over time that you can use. Okay. Another idea is to use wearable device. So this is not exactly the study that I was involved with, but when I was abroad, there are a group of people that want to understand cats, their behavior at night. Do they walk out and meet kind of the same cat? Like, do they form communities of friends? So we can attach GPS device, we can attach gyroscope, we can attach accelerometer. So you can think of putting your Apple Watch on the cat, and you can collect what the cat is doing throughout the night. We can look at how fast the cat is moving through the accelerometer. We can look at the angle, how the cat turns around with the gyroscope. And we can map the position of the cat on the map using the GPS. So this allows us to classify different acceleration and different rotation might means the cat is doing something. If it's sleeping, there would be no acceleration, no turning. But if the cat is running, then we can see it's running. So people use this kind of wearable device to understand the activities, how much the animal spent with certain action or the other. So another example, for example, if we grow the plants in different fields that we treat with different nutrients or different diseases, we can take image calculate how much the colors or the density of the rice or the wheat change depending on the nutrients or the disease. So this turns into application where in real life, we might not know the nutrient level of the plant, we might not know the disease level of the plant, the state of the plant. But when we take a new image in a new field, if we see certain colors, if we see certain density, we can learn from our experiment that maybe the plant is lacking nitrogen, for example, and the colors doesn't look as green as plants that have sufficient level of nitrogen. So a lot of people kind of rely on these kind of external phenotype to understand how molecular change or how environmental change happens inside the plant. Because you can take photo like this with less budget, with less resource than doing sequencing. Doing molecular assays give us the best kind of details, but it's expensive. So many people incorporate these kind of external phenotype because they're easy to get. And sometimes it's good enough for field application where you can have farmers take image, and from those images you can classify whether the plants have enough nutrients. But the thing is that when you design this type of application, one thing that you need to be careful of is while sequencing or molecular assays are easy to control, taking image like this, there are many things that can be beyond your control. For example, the weather. If it's cloudy, then the color is going to be different. If it's a different time of day, like different amount of light, the color will be different. And you have to make sure that you are taking photos from the same position, from the same height, like if you want to understand, estimate the density. Like, so there are many, even though this technology sounds kind of simple, there are many factors that can affect the quality or the consistency of the data that you need to be careful of. Another example, this is from my friend at our hospital. So with modern kind of AI techniques, like it creates an opportunity for you to develop your own assay. So when we learn about sequencing, like we have to buy those technology. When we talks about these videos, wearable device or drones, like we have to buy those technology. But with modern days AI, you can develop assay, you can develop a phenotyping assay as well. For example, in certain patients that have a neurodegenerative disorder, that those patients might not be able to control their hand very well. So sometimes we ask the patient to close their thumb and their index finger together, and we want to monitor how fast they can do it. That affects how much control they have over their muscles on the finger. But if you look at it by eye, right, how can you tell if this person improves or it gets worse? Right? If you look at the person moving their fingers maybe once a week, right, there's no way that you can quantify the improvement or the severity of the disease. But nowadays, because we can set up a video camera, we can use a really cheap gardening glove, and we color the tips of the thumbs and the finger and the index finger with different colors. So when we take video of this image and we ask the patient to move their fingers, we can track the position of the thumbs and the index finger automatically, and we can plot like how far apart they are from each other. So we can quantify the frequency that the patient moves their fingers, and also how close the fingers are to each other. So we get a more quantitative phenotype of the patient instead of just looking by eye and say, you did better or you did worse. So we get actual frequency as number. We get the actual movement amount in centimeters. And this can be done automatically. New patient just put on the glove, and then we start the camera, and we take the video to the software, and we can process and get the quantification. So you can create this type of assay easily with almost no budget at all. The second example, this might be more technical. So in some stroke people, they might have they may have lost the ability to move their mouth, so they cannot pronounce some sounds very well. So in clinical setting, there are certain sentences in Thai or in English that we ask the patient to say to see if the patient have a good control of their mouth or not. But who's to know, like maybe there are many different patients that struggle with different sounds. So you can experiment with different sentences that you ask the patient to pronounce. And some sentences might be very good at distinguishing a certain group of patients. And to do this automatically, like you can ask the patient to read out the sentence, like you capture it with a microphone. And inside, you have an AI model that can compare the sound of the patient to the normal people when they pronounce the same sentence, and calculate the discrepancy in the sound. So this can create an automatic assay that can detect if a certain person struggles with certain sounds or not. Okay. So these are like examples of opportunity that with AI models, you can deploy them to combine with voice data, with imaging data, with video data to create your own phenotypic assay. Okay. So once we are able to collect all the phenotype, we have the high throughput to the phenotypic profiles. How can we make sense of those data? Many of the techniques we have seen in other omics before, but we're just going to adapt them to phenomics data. So let's start with the challenge first. In phenomics, let's start with the other omics that we learned before. In other omics, the reason why data analysis is difficult is that because we have a lot of variables. We measure mutation in thousands of genes. We measure gene expression in thousands of genes. We have a lot of variables. We want to maybe predict one phenotype, one disease. But for phenomics, we still have a lot of input, like variables, and now we also have a lot of output, phenotypes. For a set of gene expression measurement, we might have the blood pressure, we might have the lifestyle of the patient, we might have the disease in the patient. We have multiple phenotypes that we have to associate with multiple genes at the same time. So that's one challenge. The second challenge is when we analyze most of the omics data, we have some knowledge about how the genes have certain function, and that certain function might relate to the phenotype, the disease that we are interested in. But for phenomics, when we take videos, when we collect behavior data, we have no idea about which gene will be correlated or be linked to the behavior or the outward appearance of the animal. We have no idea. So there is a gap in knowledge that we cannot rely on to analyze the data. And when we analyze phenomics data, we might try our best to control the environmental factor. We control the food that the animal gets. We control the habitat that the animal lives in. But there are always unknown factors that we didn't know to control. There might be certain microbiome, like bacteria inside the habitat that we have no idea that it changed over time or something else. And in some cases, we want to combine data from multiple settings, but we don't have the complete metadata information. So we cannot really exclude the confounding factor. So let's see some of the techniques that we have seen before and how they can be applied to address kind of phenomics data. So we learned about GWAS, genome-wide association study, I think way back at the beginning of the class. So for GWAS, we have a lot of genes. We have variants in a lot of genes. And we pick one phenotype that we want to study. And we pick that phenotype and we correlate with the genetic mutation. And we identify which mutation in which gene can explain that phenotype. And we have to repeat this process for all the phenotypes. One phenotype, we get one GWAS analysis. So we perform different analysis. For phenotype, if you collect a lot of phenotypes, we have a reverse analysis called PheWAS, so phenotype-wide association study. In this case, we're going to look at all the phenotype. So let's say the entire medical record of the patient. And we have genotype. We're going to pick just one variant position. So instead of one phenotype and all variant, now we're going to pick one variant and all phenotype. So this way, we want to understand how this variant can explain the combination of phenotypes of the patient. And we have to repeat this for different genotypes. So you can see how the focus is flipped. Right? In genomics, we want to understand how the genome as a whole affects a phenotype. We want how the genome affects the phenotype. But in phenomics, we have all the phenotypes. We want to understand how this combination of phenotype might be explained by certain molecular event or molecular pattern. So the focus is in the opposite direction. Okay. In the microscopy lecture, we learned about the idea of data-driven feature selection. When we analyze a microscopic image, we generate a lot of morphology features, morphology characteristics, texture characteristics, and then we use machine learning, supervised learning, to identify morphological aspects or texture aspects that can explain maybe the phenotype of the cells or the expression level inside the cell. So in phenomics, we can also rely on the same technique. Once we capture an image or videos, we can extract many information, many features, many descriptions from those videos, from those images. But we have no idea which one will be the one that are caused by the molecular details, which behavior is induced by the mutation, which behavior is induced by the microbiome, for example. So we need to do machine learning techniques to aggregate the data together. So this combines what we talked about in the machine learning lecture and the example that we saw in the microscopy lecture. We apply them to the details that we extract from image and videos, many of which we have no idea what they mean, but some of them might be explainable by the omics data or by the behavioral data. But these are not very good scientific approach. We mentioned this several times. If we do a shotgun approach where we just generate 1,000 mathematical description of the data, and we try to relate it with omics data about thousands of genes, you are bound to find some significance by chance because you have so many genes in your omics data and you have so many phenotypes in your imaging data. So maybe we need something that's more grounded in mechanisms. So people also explore how to do data-driven modeling, but do it in the scope of mechanisms that we know. So for example, we know biological pathways, we know which gene, which protein have to work together, which one is upstream, which one is downstream. We want to incorporate that structure, that mechanisms into the analysis of our thousands of phenotypes. So one technique is called a path analysis. So a path is a path. So in this case, we are going to draw a path, a network that point from upstream parameters, like the aspect of the plant. In this case, we have the plant with different growth rate, the plant with different height, the plant with different days to flowering. These are the aspects of that species. From those aspects, we can determine the biomass of the plant, how fast they grow, determine how much biomass is produced over time. And from the produced biomass, we can combine those to predict how much the plant will produce in that flower, and how much of those will turn into seed. And from this information, it might be able to tell us about the phenotype of the plant, how much it can resist certain drought, certain event like drought or salinity, for example. So using this kind of knowledge that we can gain kind of over time, once we write this kind of diagram, we can collect phenomics data. But when we are building the model, what factors we want to use to explain another factors, we're going to restrict ourselves to this diagram. So when we want to explain the biomass that we collect as a phenotype, we're going to explain the biomass using only what we know about the growth rate that we collect from the plant, the height of the plant that we measure, the days to flower that we collect from the plant. And when we want to model the number of pods that the plants produce, we're going to rely on just the biomass and the other information about the plant to use it, to explain. So we are not going to use other factors that we have no idea how it affects these parameters. So in a way, this is basically we are doing a lot of model development, but each model we are going to restrict the input and the output to be based on the scientific knowledge that we have. And this will allow us to identify the importance of each phenotype to the characteristic of the plant to this data. So for example, the numbers on the edge, these describe how important the characteristic of the plant explain the phenotype. So in this case, the growth rate of the plant explains the biomass with an importance of 0.266. The height of the plant is important to identify the biomass of the plant. But the days to flowering doesn't really affect how much biomass is produced. So these diagrams can help us identify mechanisms, relationship between the parameters in our phenomics data. Okay. So we saw this example in the generative AI lecture. So in this example, we want to understand the data. In this case, this example is an image. We want to understand what this image is showing to us. So some things we already understand. Like this is an image of a male. This is an image of maybe a Western, Caucasian person. And maybe we can even guess the age of this person. But something we don't know about this person is if this person has certain disease or not. Does it show on his face or not? So we saw this type of generative AI techniques that allow us to at least try to extract unknown factor from the data. So I want to use this example, but in the context of phenomics. So if we think about this image, this is an image that we collect as phenotypes, maybe of the animals or the plants or the patient. We have some aspect that we understand. We understand the genotype of the plant, of the animal. We understand the food, the amount of nutrients that the plants get. But there are other unknown factors. We might not know if the plant is sick or is it healthy. We might not know if the animal, what state is in. So if we follow the same technique, we want to put in our phenomics data. We want the generative AI to be able to create our phenomics data. And during the process, we ask the model to decompose known factors and try to guess what are the unknown environmental factors that we didn't include. And together, if the model can reconstruct the phenomics, which is very complex, hopefully it should be able to identify some of these unknown environmental factors that we didn't take into account. And if we want to understand what these unknown factors are, for example, we can correlate them with the phenotype that we collect. Which phenotype is produced using the known factor. Like some phenotype may be based on the age of the animal that we know. Some phenotype may be based on the nutrient level that we know. But some phenotype might be reproduced by some of these unknown factors. Even though we have no idea what these unknown factors are, from this kind of model, we can at least try to relate these unknown factors to the phenotype that they produce. So at least we know that, okay, whatever these unknown factors are, they are related to the weight of the animal. Maybe they are related to the active, the time that the animal spends doing activities. Maybe it depends on the amount of food that the animal eats. So at least we have some idea that there are certain factors about the animal that we have no idea. So knowing all these, like, what we hope to be able to do is once we have phenomics data, like, we complement that with system biology. So we understand how genotype and other molecular information explain the phenotype. So next time that we want a new plant or a new animal breed with certain phenotype, instead of doing a random random breeding, we can predict what we would get in terms of phenotype beforehand. Using phenomics data, we can train a model that look at the new genotype and predict what will be the phenotype that we get. So this can guide the way that we cross the plant and breed the animal. So people are using this similar to how we discover new drugs and new protein using AlphaFold model to predict the structure, the binding of the drugs. We can use the same idea with phenomics data to create a model that can predict which gene, which allele of the gene do we need in the offspring to get the resistance or to get the increased yield that we want. So today, what I want to show you is this kind of new other phase of computational biology. It's not molecular, but we can correlate molecular information to phenotypes that may be external to understand and to manipulate to get to improve the health of the human, to improve the health of the animal, to get a better economics, productivity out of the livestock and the agriculture. And this can involve assays that are different from what we learned throughout this course. We learned all the molecular functional assays. But phenomics also utilize imaging, videos. This in turn require modern day computer vision AI to process the data to extract the phenotype, the details of the animal and the plant from the videos and from the image. And people are exploring this kind of combination of tools and knowledge about phenotype to create application in many fields. Okay, so that's everything that I prepared for today.